right. Good afternoon, everyone. Thank you very much for joining us today and for participating in the Soil Health Training Program webinar series. This is webinar 3, The Living Soil, and today's Tuesday, December 19th. Just wanted to thank LCBP and NUEPIC again for the funding to be hosting these series. Couldn't have done it without you, so thank you very much. The agenda for today is we're going to go through a little bit of the course overview, a quick homework review, and then we have two wonderful presenters today, Wendy Sue Harper and Lisa Blazure. And then we'll go through the next class. We'll have Q&A after each presenter. And if questions don't get answered, we'll hold them till the end as well. And as always, if you have any questions that don't get answered, please feel free to ask those in the webinar evaluation form. As you all well know, this is now the third webinar, so it's 12/19. You can see that on your screen there. Our next class will be in January. We figured we wouldn't do it on January 2nd, give everybody a breath to get back into it. And so we'll see you again after today on January 19th. Sorry, January 9th. I apologize. And then just wanted to do a quick overview. Hopefully folks were able to read chapter 4 of Building Soils for Better Crops. It went over soil microorganisms, small and medium-sized soil animals, large soil animals, plant roots, and biological diversity, abundance, activity, and balance. So hopefully folks were able to read that and see some of the great visuals. But Lisa and Wendy Sue will be diving into those more today. All right, with that, I'm going to introduce Wendy Sue Harper. And so Wendy Sue is a soil scientist with over 30 years of experience in ecological agriculture education. She taught the concepts of ecological agriculture, soil science, and composting ecology for Prescott College, Green Mountain College, Hogback Community College, the University of Vermont, and Dartmouth College. Wendy Sue teaches soil science and garden planning for the UVM Extension Master Gardener Program, basic composting and soil science for the Vermont Master Composter Program, and ecological soil fertility management for the UVM Farmer Training Program. She has conducted farm, processing, and compost audits for the Vermont organic farmers since 1998. And Wendy Sue also serves on the board of directors for the Composting Association of Vermont. With that, Wendy Sue, if you'd like to share your screen and get going.
Thank you so much for having me. All right, we'll just put this in presentation mode.
Looks great on our end.
Wonderful. This is the first part of Living Soil. Lisa will do the second part, and we're going to talk about organisms and connections. We really want to talk about soil biology for you folks to help you understand its importance, its relevance to healthy soils and growing healthy plants and healthy ecosystems. To start us off, I want us to think about three concepts: scale, function, and connection. And I'll have a slide for each of these. And a reminder that all of this happens all together. So for scale, we think of soils as being this, this like terra firma, dry land, but within soils, there are these multiple worlds based on scale. And there's a watery world that exists within them. And I'll start on, excuse me, on this slide with the bigger organisms. We have big organisms in soil, like insects and worms, that make pores. So these are the organisms like earthworms that drill down and make pores to live in. And then we have medium-sized organisms like mites that walk knee-deep in water in these pores. They're tiny. Those we can see. We can pick up earthworms and hold them in our hands, but the mites we can see with our eyes, but they're small. And then we have these very small organisms that are microscopic, that are swimmers. And these swim in the thin skins of water that coat soil particles and aggregates. And so we have these three worlds within soil. And we're going to look at organisms in each. So then for function, organisms do many things in soil. They decompose plant and animal residue and release nutrients and make humus in the process. They can transform nutrients into forms that the plants can use that are available to them. They can improve soil structure and tilth by number one, and improve plant nutrition by all of those things above, by facilitating nutrient absorption. Diverse populations increase biological stability and balance. And that means that you have a resistance to system degradation, and the system can bounce back faster when you do have some kind of incident. And also, we get what ecologists call emergent properties. So these are properties that help the system create self-sufficiency. And that includes things like nutrient cycling, beneficial organism relationships, and then pest management, pest population management, and disease suppression. And an example of that last one, the disease suppression, we're going to talk about today later on. And if you think of something like a forest ecosystem, the northern hardwood forest, if you took all the invasives away and the problems we've created for them, you have a very self-sufficient system that does this naturally. Now, I want to mention that the picture on the right at the top is actinobacteria. It's a type of soil organism that we'll talk about. And then the one on the bottom I put in there because this shows that microbes, and this is agar plates that have either zinc phosphate, magnesium phosphate, etc. on them. And you can see there's a halo around the bacteria colony. And that's because they can dissolve minerals too. They're able to do that and get the phosphorus from the agar. So this is a really beautiful diagram of soil organisms of soil ecology produced by Dr. Daniel Dindal. He's passed now. He was in SUNY Syracuse for many years, and he produced this for the compost community in 1979 when they asked him to create something that showed the web of life. Now, many years later, he said this actually represents all soil too. This is a really simplified diagram though. And each organism can represent 100 to 1,000 species. And not all species are even on here. There's many other types of organisms as well. And UVM Library has his two 800-page volumes of his life work that have descriptions of soil organisms in the library if you're interested in digging in a little deeper. I like this diagram though because it shows many things. Number one, you see first degree, second degree, third degree. Those are the trophic or feeding levels. So you have organisms that eat the waste directly, primary decomposers, secondary eat the organisms eating the waste, tertiary eat the organisms eating the waste. So it gives you an idea of how things move through the food web. Also gives you something about scale because it has the millimeter size of these organisms. And then lastly, it shows the foundation of the food web is food. And I put a red circle around that. If you're going to have soil biology, you have to have food to feed them. They won't be around if you have no food. And, you know, the system isn't completely efficient. At each level, you have some heat loss that happens as you go up this kind of food web or food chains. And just for people interested in compost, that loss of heat is what's captured in the compost pile that comes from the microbial decomposition of waste. So let's look at some organisms. We're going to start with bacteria and archaea. And these are both considered microflora, but they have their own kingdoms. And remember, these are swimmers, so they swim. And hopefully the image on the right shows you the bacteria with these whip-like structures coming out its end. That helps it swim. So these are swimmers. They do move around. So anyways, bacteria are very common in soils. They're very tiny microscopic organisms. To see them, scientists grow them on agar plates, which we have in the top left. You can see the colonies grow, and each colony would have 100 bacteria in them so that you can study them. Now, bacteria and archaea, they're single-celled and they come in different shapes, rods and spheres, and they look very much alike, but they're a little bit different with structure. And they broke the archaea out because when they looked at the cell walls and membranes, they found that, they've got different compounds in it. So as we dig deeper into structures, we tend to break things out. The one thing about the archaea is that they tend to exist in really harsh, very salty environments. They can produce methane, so environments that have that, and very hot environments as well. And they haven't found any that are pathogenic at this point. Now, for the bacteria, they're very common in garden and farm soils. They like a pH between 6 and 7. There's 100 million bacteria in a pinch of active farm soil, and more bacteria than number of people on Earth in a handful of active farm soil. So there's a lot of them. And they're really important to decomposition of carbon compounds. That's their energy source. So they break them down to get energy. And they're very important to plant nutrition because they do things like run the nitrogen cycle and the carbon cycle. And they also can exist in environments that have oxygen. We call that aerobic. And we say anaerobic, it's really anoxic. It's environments without oxygen. And a lot of bacteria are actually facultative anaerobes. That means with oxygen, and they're fine with no oxygen, which is why compost piles can, if your management isn't quite good and it gets a little too dense or wet, it can flip to anaerobic decomposition, which is stinky and produces toxic compounds. And that's because it's the same bacteria doing the same thing. Okay, now, let's see. That's it. Go on. Another type of bacteria, and this used to be called blue-green algae, but it's not. It's a bacteria. And so, like I said, as they study things more and more, they move things around. This is a cyanobacter, and it produces its own food, and because it contains chlorophyll. And so they are really a surface phenomenon in soil, things that are phototropic. And then here we have actinobacteria. Now, these used to be called actinomycetes, but they renamed them. And they're kind of beautiful. They grow in coils. They kind of look like they're halfway between fungi and bacteria because they do grow in coils. They produce, you can see some images of that. They produce spores, and the bottom shot is a reproduction structure where the spores would reproduce. These guys like aerobic warm soil or compost, pH of 6 to 7. They do not tolerate acid environments. They're very good at decomposing kind of tougher materials. So think cellulose in the veins of leaves, that kind of thing. Another really special trait of these guys is they produce a compound called geosmin, and that compound is what makes soil smell like soil in spring. That amazing spring scent that you get, that's produced by the actinomycetes. So when you smell it this coming spring, you can think, oh, the actinomycetes. And if you plate it on agar plates in the lab smells like that. It's amazing. There's no soil around, but the lab smells like spring. And that compound can also be found in things like beets and wine. This one is the genus Streptomyces, which sounds like streptomycin. And I want to point out that there are a lot of antibiotics that come from soil organisms, not only bacteria, but also fungi. In fact, half of all human antibiotics come from actinobacteria. So they're really important for us to help us defend ourselves. They use it to defend themselves and stake their claim. And a lot of these things have been discovered by soil biologists. So we're going to move on to mold. We're still in the swimmers category. So these are microscopic, unless you grow them out. These are molds and fungi, and they're considered microflora. They have their own kingdom. And fungi includes molds, mildews, rusts, smuts, yeast, mushrooms, puffballs as well. They're very versatile and persistent in soil and can decompose some tougher stuff like woody materials. In fact, a lot of composters will if they get in horse manure that has a lot of bedding, they'll let it sit for a few years to let the white rot fungi get at it before they use it in their compost recipe. Fungi produce spores. These are like fungal seeds. And you can see the bottom two slides over to the left and middle show an actual picture of spores and then these beautiful structures that produce them. And they have thread-like structures called hyphae. and they make up more biomass than many other groups. They tolerate acid conditions. They're very important to decomposition in forest soils, especially acid forest soils. And the mycorrhizae, which I'm going to talk about a little later, and I know Lisa is too, are really essential to the formation of soil structure. And I'll talk about that. Now, the left picture, the mold, that's pure culture penicillin, which has also been very important in human medicine and human history. And the bottom right, you see the halo around this colony, and that's because there's antibiotics being produced. They're protecting themselves, and the fungi aren't going to grow any closer to that because of the antibiotics that are there. The fuzzy structure you see on a couple of these slides are the fruiting bodies, and that produce the spores. And if you see that in your refrigerator, you should never smell it, because you will get hit with more spores than you ever get if you're gardening or doing anything else. And there is something called farmer's lung. It's an allergic reaction to the fungal spores in moldy hay dust, and it's a terrible occupational disease. And toadstools or mushrooms are also another type of fruiting body. These are not the only ones. Okay, so now I'm going to show you. Oh, I also wanted to add that many molds produce defense compounds too, which we call mycotoxins, which can be harmful if they're fed to animals or in the animal feed. Okay, so this is a little video clip showing fungi, hyphae growing toward each other, and then they stop. And they stop because they sense each other because of defense compounds in the soil. Okay, let's see, we go to the next one. Okay, so here are some other examples. Fungi can be saprophytic and they can be parasitic. And saprophytic one is, you can see the hyphae growing through a blade of grass, and the parasitic one is invading an amoeba. Now, dampening off is, which some growers who are in greenhouses have problems with, is also an example of a parasitic fungi. Okay, I also wanted to mention here that the largest organism on Earth is not the blue whale, but a honey mushroom fungus discovered in 1998. It's estimated to be over 2,500 years old, and it covers about 2,500 acres in eastern Oregon. So fungi are a really important category. They produce really strong compounds, these compounds that stick around. They're carbon-based rings. They're complex structures. The name benzoic, these things are carbon-based. This will not be on the test. And when you extract them, you extract from fungi, you can see, so we're starting at the top left and working across the top, and then we'll work across the bottom, left to right. So the top one, you show the extract, can be extracted from fungi. When you grow the fungi on agar plates, you can see these melanin granules there. Those complex structures are there. And you can, when you look at the fungi itself, you can see it in between its cells. When you destroy the fungi, you can still see these granules. And the last shot, when a nematode eats them, it's still inside the nematode. So these things stick around. They're very complex. And this is really the beginning of humus. So we're going to also talk about microflora that are in the fungi category. Yeast. These are single-celled organisms. They reduce by budding. They don't have hyphae. Well, most of them don't. There's so many categories in between. I've really simplified this for today. They're involved in fermentation, have a modest effect on decay. So now we're going to go to another microflora. These are algae. And algae are also photosynthetic, like the blue-green algae, which are bacteria. They have their own kingdom, a real algae kingdom. And in compost piles or soils, again, this is a surface phenomenon. They need light in order to do their processes. So you might find them on the surface of the soil. And you might have seen soil that looks greenish on top where we have algae. So they're producing their own energy, and they're considered consumers. And they're kind of a minor organisms in the process of decay. Oops, did I miss one? Hold on. Yes, I did. Okay, so now we're going to go to microfauna. So these are organisms that we consider more animal than plant. And we're going to look at protozoa first. And again, these are swimmers, and you can see in the top right photo, you've got that whip-like structure that helps organisms move. Protozoa have their own kingdom, and we've got three types here, and that I'm showing you. One is the ones that are the amoeba, that's the bottom right, that moves around with a fake foot and engulfs prey, and I'll show you a video of that. You've got ones that have little hairs, ciliates on them, and they move that way. And then you've got ones that have flagella that help them move. That's the top left one. And then you've got some that are actually tied to something, and those are the vorticilia. So they are tied to some structure. They're not free living essentially. And protozoa feed primarily on bacteria, but some eat other protozoa, organic matter, sometimes fungi. They have really been overlooked in the past. And I think Lisa's going to talk about this more too. But they're especially important to nutrient cycling. Okay, so this is the amoeba one. This one you can see the bacteria, and the amoeba is going to move and they engulf it, and it goes right inside of them. And then this is paramecium doing their little lives. And they also are eating bacteria. So the next organisms we're going to look at are rotifers, and they are predators and scavengers. They consume bacteria, protozoa, fungi, and algae. They're found in water. Most species are found in water, but also in moist soil. And they go into a cyst state if the soil dries out. And this is true of many of the microorganisms. If they don't like the conditions, they'll go into a senescent state and wait for the conditions to improve. These guys can be an indicator for pesticides and metals at sewage treatment plants. They have a modest effect on decomposition, but they look wonderful. So now we're going to go on to nematodes. And these are also called eelworms. They're found in soils everywhere. Arctic, deserts, everywhere. They eat many things and have mouth parts that fit what they eat. So mouth parts for feeding on bacteria, fungi, protozoa, algae, other nematodes. There's a very small number that are plant parasitic, but unfortunately, that seems to be the focus of nematodes. The mouth parts do tell them apart, and if you look at the bottom one, the one on the right has this lip-like structures that help them grab onto bacteria. And the one on the left, excuse me, has like a hypodermic needle. Now, plant parasitic nematodes have these, so they can go into plant and suck the juice out of the roots. But also, nematodes that feed on fungi have these, so that they can eat the fungi as well. This can be a problem in some places, like in Vermont, like old apple orchards have problems with lesion nematode. It is a part of the apple replant disease complex. So still mesofauna, and these are the ones that walk knee deep. Or actually, we're moving to mesofauna. These are the ones that walk knee deep in water. You can see these with the naked eye. So we're moving up a magnitude of size. And we'll talk about mites, and all the things on this slide are mites. Mites are really critical in the soil food web as decomposers and predators. Now, one of the things they do is they break up larger pieces of organic matter. They break them apart. They break them down so smaller organisms can have access to them. Somebody has to do that job. And they also disperse bacteria and fungi in their waste. So the mites we have a mold mite, and they eat yeast and fermenting organic matter. These can actually be a pest in fermentation industries. They can have problems with them because they can tolerate anaerobic conditions, or conditions that don't have a lot of oxygen in them. The beetle mite, which is up on the left side top, feeds mostly on fungi, but also nematodes and debris. Predatory mites, which are the bottom slides, eat nematodes, springtails, other mites. They often are seen in spring in compost or and they can be amber in color and about a 1/25 of an inch long. And when I used to teach composting at the University of Vermont, we would put a white cloth down. It was a spring class, and spread out the compost, and we would find them because you can see them against the white background. So here's another wonderful video that shows everybody doing stuff all together. Soils are really very active. Now, another mesofauna, so remember these again, walk knee deep in water. We have bark lice, which eat vegetative debris. These are kind of cousins to book lice that can be a problem for libraries sometimes. We also have springtails. Springtails tend to be funginiferous. They mainly eat fungi, but they'll also eat algae, nematodes, and debris. They graze fungi and algae like cattle graze pastures. And as decomposers, they have a huge impact on nitrogen mineralization and plant growth. They also can be selective in what fungi they eat, and they can alter fungal communities because of this. They're used in Europe as indicator species when they do pesticide testing. Scorpions, that's the bottom left. They're not in the scorpion family, and they don't sting. These guys walk around with these big claws out in front of them, and they feed on small insects we call microarthropods, like mites and springtails. They have no eyes and no ears. They use vibration to sense their prey. And then the beautiful featherwing beetle, which I've never seen, eats fungal spores and rotting litter, and they're supposed to be abundant in compost and manure. So now we're going to go on bigger macrofauna, the poor makers, and we'll start with earthworms. And a lot of these organisms, I'll just say this in general, these organisms are kind of soil engineers. They create pores, they move soil around, and they're not the only ones who are soil engineers. I'm thinking ants just popped into my head, but they do make pores, and they move soil to the surface and allow water to go in. We'll talk about earthworms, and they need a soil that's moist with a temperature of 32 to 86 degrees Fahrenheit, pH of 6 to 7, so not too acidic. So food. If they don't like their conditions, they burrow down and kind of wrap themselves up and just wait until they improve. And they have a gizzard-like structure that grinds things, grinds soil particles, and they have microbes that are partners in their gut, which makes them very efficient at breaking down organic matter. They can digest a whole acre of soil in a year. And their casts, which pile up at the top of their hole, have much higher nutrients like nitrogen, phosphorus, calcium, potassium, and even cation exchange capacity. So than the bulk soil. So they do increase fertility. Their bodies will absorb pesticides or metals at the concentrations that are similar to the soils they live in. They produce sticky substances which help glue soil particles together. They line their burrows with that. And they collect weed seeds and stick them in the burrows, and then they eat the fungi that grows on them. So they're like little agriculturalists growing fungi. And their channels improve aeration and are pathway for water into soil. Now, about 1/3 of the worm species in the US and Canada are not native. And non-native night crawlers, we're not even talking about the jumping worms. These are night crawlers, have created problems in some forest system by eating the organic matter. Red wigglers, which people use in earthworm composters, are non-native from Europe, and they're now found on every continent but Antarctica. And they are highly adapted to decomposing organic matter, which is why they're used in earthworm bins. They have the potential for being invasive, but people really haven't found them being invasive so far as I could tell. So not yet. And we'll talk about the jumping worm in the next slide. Now, white worms or pot worms are a whitish kind of translucent cousin of earthworms. They can take wetter conditions and soils that are a lot more acid. They eat bacteria, fungi, and soil organic matter, and often used as an indicator species for soil contamination. Millipedes, which is the bottom slide, are significant primary decomposers. So they feed on the waste directly, and scavengers. They fragment 10 to 50% of the annual leaf litter fall, and facilitate microbial decomposition. They moisturize their foods with secretions. They kind of scrape it with their jaws, and they're very slow moving. And these are not the ones that sting. Centipedes are the ones that sting. People mix them up sometimes. So now we'll talk about the invasive worm. And so, and this I got from a website that the state of Vermont has. Jumping worms are an invasive earthworm. There's several species that make up these. They're established in Vermont and appear to be spreading recently. They rapidly consume organic matter, deplete soil of nutrients through leaching or erosion, and they also can damage plant roots in nursery and produce operations. The jumping worms and their cocoons, and next to the penny shows the tiny little cocoons, those kind of like their eggs, can be spread through compost, leaf litter, wood chips, nursery plants, and soil. So you need to have protocols with the people that you're working for to try to keep these things out of their farms. The adults will die off over winter, but the cocoons don't. And when they go through organic matter, they eat all the organic matter and leave the soil looking like coffee grounds. And there's maybe some other issues with them too, but they're not good and you don't want to get them. One of the definitive characteristics, a regular worm, and I'll go back a slide, you can see the bulge. Can you guys see my arrow? There's a bulge.
Yes, we can.
So there's the bulge. That's the structure. That's regular worms. The jumping worm, this thing is flush, completely flush. It doesn't stick out at all. And they move kind of like a snake. Okay, let's see. Our next one, our macrofauna, include, and these are more pore makers, the isopod, pillbug, sow-bugs, also called wood lice too. Very good at breaking down tough material. They're considered primary decomposers as a result, and they like dark, moist area because they lose moisture very easily through their cuticle. I'm going to talk about snails and slugs together and then a little separately. They are decomposers and scavengers. They live in moist litter and soil, and they feed on fungi, decaying plant matter, feces, carrion, fresh plant material too. Predatory snails and slugs feed on earthworms, potworms, and other snails and slugs. So you can have them eating both plant and animals. Some also will eat live tissue in the garden and can be a pest. And they have a moderate impact on decomposition. Now, for land snails, these are important food in soil ecosystems, and they actually help move calcium around the ecosystem too. And there are at least 70 species of non-native snails in the US. For slugs, they leave kind of slimy trails, but the trails actually have fibers in them that help them hold on as they climb up plants. And they come out at night or when it rains because they need moisture. And the Northeast have 3 native slug species and 13 introduced species. So we have some issues with that as well. Now for maggots, and these are fly larvae, in their immature stage, they do such a service by eating dead things. So we're not up to our eyeballs in animal remains and manure and things like that. They're really important and recyclers. So a few more macro pore makers. We have ground beetles and centipedes and ants. And I'll start with the centipedes. They are mostly predatory of other invertebrates that are at least that are their size or even larger. And they're very fast moving, and some of these species will actually sting people. And for ground beetles, there's a lot of beetles, but we're going to talk about ground beetles. They eat other insects and vegetative debris. Studies at the University of Maine show that the loss of weed seeds after what they call a weed seed rain, this is when you have a weed problem, you don't deal with it. And the ground beetles are really important to eating the weed seeds. In fact, you can create areas called beetle banks on farms that house these guys to help them do that. Now, ants are one of the most significant insect groups in soil. They move a great deal of soil and influence soil structure. They are important seed dispersers. They're generalists, Scavengers eat plant debris, insects, uh, and snail eggs, earth from cocoons, that kind of thing. And they pull waste into soil, helping to granulate it. So they have an important impact on soil aeration and infiltration. They stimulate the nitrogen cycle, and they use pheromones to mark their trails so they can follow where they've been. And then our last microfauna is for pore makers are tubellaria, which can be from a 1/2 inch to 12 inches long. These are of earthworms, and they eat other small invertebrates and protozoa. Some of this group can be parasites. And then we have spiders, and this is the wolf spider. It doesn't make webs, and it carries its egg on stacks and babies actually on its back. It takes care of its young for a while. They don't have a flashy appearance, and they depend on camouflage for protection. There's many other insects, beetles, bees, wasps, as well as turtles, owls, and gophers that live in soil. We don't have time to talk about all of them. So for macroflora, we have plant roots, and this is the largest macroflora. Plants have a major impact on soils physically, chemically, and biologically through their growth and development. And they have their own biome. Plants have their own biome, just like people do. And this is a root tip of a plant with a plume of mucigel around it. And the rhizosphere is the part of the soil that's in direct contact with the plant. And it's in here that the mucigel makes the root have contact with the soil, and it's full of wonderful organic compounds that bacteria and organisms eat. And as it pushes through soil, it sloughs off cells, and it also allows the cells to be consumed by bacteria. Now, so we talked about the root tip on the left. On the right, we show plant roots with little bacteria on them. This is also an image of the rhizosphere. And in 2015, Science News had this really interesting article talked about how plants shape their rhizosphere, their microbiological communities in their rhizospheres by using defense hormones like salicylic acid, jasmonic acid, and ethylene. They don't completely understand it, but there's this thing going on where plants help create who's in their rhizosphere. And here's a nice video of a plant root moving through soil. And you can see cells sloughing off. And here's another one of roots growing in soil. And you can see the cytoplasm moving inside the roots. And there's a little bacteria on the outside. Sometimes bacteria exist inside the roots too. They go inside of there. Whoops, we're gonna go. So our next part, we're gonna talk about connections between plants and organisms. And we're gonna start with the nematodes, which is those thread or eelworms. There's really one interesting relationship where when the nematode feeds on this specific fungi, it makes fungal nooses. And you can probably see the little nooses on this bottom slide. And up here, it captures. And when the nematode swims through it, it captures them. And then the fungi eat the nematode. And you can see the hyphae coming out of this nematode. And so we have populations, checks, and balances between these two organisms. And Bill Murphy's, one of his last students, was trying to look at how could you could make this fungal thing work to help with parasites on pastures, but I don't think they ever didn't get too far with it, but they were looking at it, and it's a good idea. And here's a nice video of there's the nematode. It swam into the noose, and oh boy, it's stuck now. That's the end of it. So another interesting relationship has to do with the movement of decomposers in ecosystems, because, how do you get these small organisms from site to site? You think, especially if you've got, the prairie and buffalo roaming around, and they'll poop in one place, they'll go 5 miles and poop someplace else. How do you get them where they're needed? And how you do it is through foresee. And it's a non-parasitic relationship where you have detritivores hitchhiking on organisms. And Daniel Dindal did this with flies, but the image here shows a hermit flower beetle. This is the top image on the right. You see all those little white things. Those are immature mites, and they're between the head and thorax of the beetle. And they have a sucker dish that allows them to jump on and stick. So they can stick to the surface of the insect. And on the mites are nematodes. So they're phoretic on the nematodes are phoretic on the mites. And so how this works is that Dindal found that flies produce a chemical that is attractive to the mites. And the mites in their immature stage climb on the back and off they go. And they go to a food source and they're eating the food source, and the mites also eat nematodes and they eat the fly eggs too. And they can sense ammonia, and when it decreases, they know it's time to hop on in the immature stage, hop on another insect and go for another ride. And it's not just flies or a beetle like the hermit flower beetle. It's also pitcher wing flies, humpback flies, fruit flies that will do this kind of thing. So it's like transportation, and it's not a parasitic relationship, or not considered that way. Mycorrhizae are one of the most important organisms that we have with plants. And about 80% of the world's plants actually have them, a lot of legumes and cereal crops. And there's two main groups, but there's many, many types. So we have the ones that grow on the outside of the plant, and those are ectomycorrhizae. You can see they look like little hands. And those mostly form on trees. And there's ones that go right inside the hyphae, go into the cells, and there's exchange of nutrients. And those are called endomycorrhizae or VAM, versicular arbuscular mycorrhizae. So, and there are some that actually are kind of in between these two. But what they do is they help plants grow better. And on the bottom left, you can see trees with and without the mycorrhizae. And in the middle, you can see and without the mycorrhizae. They need these infections. They're really important for their nutrition. And they think that the ectomycorrhizae protect the root from diseases because they have to go through the mycorrhizae to get to them. And the endo help with a lot of things like water, drought resistance, and nutrition by extracting things like phosphorus, because they increase. And if you look here, you can see the gold is the root, and all this other stuff is the mycorrhizae. They increase the contact with the soil 100 to 1,000 times. They even help with nitrogen. One study, they were studying springtails and feeding them mycorrhizae, and they kept dying. And they realized the mycorrhizae were poisoning the springtails. And when they radiolabeled the nitrogen in the springtails, they found that 25% of the uptake of these trees of nitrogen came from springtails, and the mycorrhizae were mediating it. And again, this is not a parasitic relationship. If you have high phosphorus soil, sometimes these relationships won't form because the plant doesn't need them. And some plants don't host mycorrhizae. This includes the brassica family, like cabbage, broccoli, mustard, turnip, canola, etc. The amaranth family, beets, chard, spinach, quinoa, and amaranth, and then the buckwheat family. And they're really important in soil ecosystems. And the other thing about mycorrhizae is that they make connections. It's not just, I've been pretty much talking about, agriculture crops. Now, actually, I'll talk about this later. I'm going to go to the next thing, and then I'll bring this up again. So another great relationship that's really important for agriculture is rhizobium and legumes. And this is a bacteria. When the bacteria touches the plant root, the plant grows a fleshy nodule around it. So this is the plant growing something. It is a lock and key kind of effect. And the thinking is that the enzyme nitrogenase that allows these bacteria to take nitrogen out of the soil and put it in a form the plant can use is poisoned by oxygen. And so the nodule protects it. And if they're pink, and you can see the pink picture on the right side, they're working. And if you have legumes fertilized by nitrogen, they don't really form, or to a lesser degree, they'll form these nodules because, again, it's not a parasitic relationship. In pasture studies, they show that if the grass is grazed and this would be a mixed sward, is grazed too short, the legumes will drop their nodules. They'll pick them up again when they can carry them and feed them. They won't have them if they can't make it work. So there's that. And then here, so this is the one where we show the connection between plants. This is corn and soybeans. But they have done studies and they found that corn that's connected by mycorrhizae to legumes has more nitrogen in it. And in forest systems, we know that they're connected underground between trees by mycorrhizae networks. And, you can have a mycorrhizae network that has several tree species on it, and you can have different networks. And so two trees may not be on the same network as well. And that helps feed small trees and trees that are in shade. It helps them in the research to do that. Anyways. And then here, this was discovered it was published actually in 2018, where they found this indigenous corn. These are aerial roots of corn, and you see the mucil gel on there dripping off of it. That's loaded with free living nitrogen fixing bacteria. And they determined that somewhere between 29 and 82% of the corn nitrogen came from this microbial association. So, people have always thought, well, wouldn't it be great if corn could be made to make its own nitrogen? Well, there's an indigenous one that came from Mexico that does. And then another interesting relationship is the rhizobacteria, these plant growth promoting rhizobacteria. They do many things for plants. They fix nitrogen, they promote growth, they help with solubilizing phosphorus and iron, and other nutrients. They control pathogens, they reduce stress. And there's actually communication between the microbes and the plant that goes back and forth with chemical signals. And so they help with heavy metals, pollution like hydrocarbons, salinization, and drought. And then we also, and this is almost my last one, I want to talk about disease suppression. This is something that happens in the root zone. And it started out with compost in the 60s. Rhododendron growers found that when they put compost in their potting mixes, all their diseases changed. And they got a plant pathologist, Harry Hoytink, involved in it. And they now know that almost any organic matter addition does this. And what it does, it stimulates the antagonist. So you have things like Bacillus, Enterobacter, Trichoderma, Streptomyces, and Pseudomonas, and more. And Trichoderma, you might recognize as root shield. It's been commercialized too, and people will put it within their potting mixes sometimes, or actually in the soil. Vegetable growers will do that. They know that these antagonists, single strains, are not as effective as mixtures of microbes, and how they do it is these microbes produce antibiotics that harm pathogens, and plants will actually take up some of them and use them in their defense systems. They will outcompete the pathogen for nutrients, or they will directly kill it. We call that parasitism. And they also can, and Fred talks about this in chapter 8 of his textbook, they can cause induce systems resistance or systematic resistance. It's like a plant vaccination. And Fred talks about there are actually 2 types of those. But not all diseases are suppressed. Some are easy, some are had, and it only lasts 6 months. On my last slide, which came from Fred, actually I have 2 more, is about, let's go up a level with connection, 1 more level. So this shows that plants, when they're attacked, they produce defense compounds that are volatile, and they'll send them out into the world. And so they'll do this, and beneficial insects can read them, and they produce different stews of compounds depending on who's attacking them. And they also, at the same time, will produce compounds that give the insect a stomachache, and it slows their feeding. And plants will produce nectar in places that aren't a flower. These are called extrafloral nectaries. And a lot of plants have them. You probably know that peony, the bud covers, they always have ants on them. And that's because they have extrafloral nectaries in the bud covers, and the ants are looking for the sugar. So anyways, it's a 5-star hotel. The insects come in, they lay their egg, they get some food, and off they go. And there's this real connection chemically between the insect and the beneficials. And last, we know that animal saliva stimulates plants to grow. And it's not just sheep and cows, but also I saw studies on moose and on goats and other animals. So there's a connection of animals to plants, an evolutionary connection where that saliva, when they eat, those chemicals will help the plant grow or stimulate the plant to grow. That's it. Any questions?
Wendy Sue, I am completely impressed by how much knowledge you just shared with us. Thank you so much.
I'm sorry it was a little quick.
No, that was great. If you'd be willing to stop sharing your screen, we
Yes.
can
Oh,
see a
I'll do
couple more
one more
folks'
slide. That's my
faces.
biological, my references that I've got that I used. Most of the slides came from them. All right.
Wonderful. We have one question in the chat from Mel. Do the pest suppression benefits of soil health also apply in a forestry context?
I believe, yes, it's all plants. It's not just agricultural crops that they do it, because, and that would be in a forest that has organic matter because it starts with the breakdown of organic matter to stimulate these organisms to develop. So those antagonists, you need a biologically active soil is what I'm saying.
I've got a question from Sarah. I was wondering if you could speak about whether it's worth using general mycorrhizal fungi that is sold in stores to inoculate soils.
So I think if I would say, so I have a lot of thoughts on this and I'll share them. First of all, I think that we have been very casual at introducing new things to our systems, like new species. And years ago, I did a talk, I was worked for No for Vermont. We were having these people come in and talk to growers. I was organizing it. I was not the speaker, the organizer. And one of the people from Cornell said that if you look, what you find with, you're talking about rhizobia, is you don't find the ones you've inoculated with, you find the native species. They have hard, and then I tried to pin her down on it and she's like, well, she kind of said no, but she said it. I heard her say it. So I would be very careful at if you have never grown beans or peas before, you probably do need to inoculate with some rhizobium. But I would be very careful with what I put for organisms out in the soil, because we keep having these things like, oh, now we have invasive snails or whatever, we have invasive worms. And so we haven't been as thoughtful as we can be about doing that. So that's number one. Now, if I had let's say we had a site that was stripped of all topsoil and everything, then maybe there's a role for them in some places where you have really infertile or brutalized soil, toxic waste places or something like that.
Thank you. Yeah, I've heard too of people bringing like rice into the forest to try to get that inoculated with the forest fungi and then bringing that back out into the agricultural fields. Not sure if that's a good idea or not, but
Oh,
lots of
interesting.
options. Yeah. We've got another question from Isabel. After an intense event such as flooding causing erosion of the soil layers, how does the soil become biologically active again?
Oh, that's a great question. So I'm assuming you mean a farm field soil, and not a forest soil, because that is pretty much which is along rivers. And so we have this disturbed soil. We disturb the soil along our river systems, and because they've been tilled, they have they can be impacted by organic matter. I was doing an organic inspection in the Intervale 20 years ago, and they had a little flood, and half the field was tilled and half wasn't. And the farmer said, Wendy Sue, you got to come and see this. The one that was tilled had a crust I couldn't break between my two fingers. And I'm not a small person. I'm pretty strong, and the one that wasn't tilled was fine. So, some of it is caused by that. And water weighs something like 64 pounds per cubic foot. So that's a lot of pressure on soil when you think about flooding. So the first thing I think, and some of this I know the state of Vermont tells growers, you want to dry it out before you get your equipment in there, because there's a lot of crap in flood water that, is not good. anything that can get picked up, road oil and salt and everything goes into that floodwater. So you let the stuff dry out and then you till it. And the way to, I think, make it biologically active again is to add organic matter. And by that, I mean, add it, but do it within, if you have high phosphorus, add organic matter that doesn't have phosphorus in it. So you're not increasing a problem. If you don't have high phosphorus, you could add something like compost that is like a bioinoculant that has some organisms in it that can help kind of bring that part back. But the whole thing is the foundation is food. So if you've got food, you're going to have organisms there. And remember, a handful of active farm soil has more bacteria than people on Earth. So it's already there, You think about how many of those are on a head of a pin. It's just amazing how active life is. And so it means, give it some food, bring it back that way. That's what I'd recommend anyways.
Thank you. I had one question. One thing you mentioned about the fungal phenol compounds, that's what forms the humus. I wonder if you could just speak a little bit more about that. I think that kind of theme has come up several times in past webinars.
So we know that humus is ring structures. Well, we used to think it was like only big ring structures, but now people have done more work and there's some smaller, as we look into stuff more, we can say less about what it really is. We used to say it was only these complex ring structures, high in organic acids and high CEC dark color. That's how we defined humus for decades. And now we know, well, maybe it's not all big structures. Maybe there's some smaller structures in there as well. But ring structures are the beginning of making humus. And lignin also has ring structures in it. So, if you have woody compounds, if you can get the white rot fungi to start breaking them down, you can also get really great humus from that as well. So, did I answer your question? Or ask a more detailed one if I didn't?
I think I just wanted you to talk some more about it, and you did.
Yeah.
So I think you nailed it. Thank you very much. And I think two things that really stuck out to me from your presentation before we transition to Lisa was the idea that soils with too much phosphorus might not form the mycorrhizal partnerships for the
Or not as
plants.
well, right? Because it's not parasitic.
Right. Um, and then that in pastures that are overgrazed, the legumes might drop their nodules. That is so interesting.
That came from Bill Murphy's research or his research group that, it's not a parasitic relationship. So if they, and it's also that they've got to have the oomph to feed them. If they can't take care of the mycorrhizae, they'll let them go and then they'll pick them up if you graze it really too short, and then they'll pick them up again and start the relationship again. So yeah, that's really interesting the way it all works.
Thank you so much, Wendy Sue. Oops, sorry, go ahead, Willie.
So, hi, Wendy. So good to hear you. What a great presentation.
Hi, Willie. How are you doing?
It's also interesting, too, that the rhizobium won't function if there's a little too much nitrogen of available sorts, especially nitrate in the soil. So we can over apply and then legumes will use it, but they will not fix any nitrogen while that nitrogen is available.
Or they'll fix less. They won't have the same extent. Yeah.
Wonderful. Thank you so much. I do want to invite folks, if they have more questions for Wendy Sue, please put them in the chat. We're going to transition over to Lisa. But if folks still have questions after both presentations, please don't hesitate to put those in the chat. Thank you again, Wendy Sue. All right, now we're transitioning to Lisa Blazure. Lisa is an experienced conservation professional who advocates for improving soil health to achieve water quality goals, grow nutrient-dense foods, and enhance farm profitability. She works for Stroud Water Research Center and serves as the coordinator for the Pennsylvania Soil Health Coalition to foster collaboration among numerous stakeholders involved in soil health education and research. Lisa networks with a broad range of colleagues to implement conservation practices and organize educational programs. Prior to joining the Stroud Center, Lisa was the agricultural technician for the Clinton County Conservation District and worked in the Chesapeake Bay and nutrient management programs. She also serves on the Pennsylvania No-Till Alliance Board and the Pennsylvania Sustainable Agriculture Board. With that, Lisa, if you'd like to take it away, the floor is yours.
All right. Happy to be here, everyone. If you need to get up and stretch a little bit after Wendy Sue's marathon stretch through the soil microbe, go ahead and get up and stretch a little bit here. I'm going to start my talk. So Lauren, let me know when you're seeing things that look okay on your end.
That looks great.
Okay. All right, so we are going to dive into part 2 of this soil biology session. And as you can see by the breadth of what Wendy Sue just covered, it's a really kind of complicated, complex, and we're learning so much about this biological system in our ag soils. It's amazing. And that's really kind of where that new kind of cutting edge frontier is on these conversations around soil health, is really learning about the intricacies and the ecology that's happening in these soil structures. All right, so for my part, for part 2, we're going to touch again on soil organic matter levels. I know Fred Magdoff covered some of that last week, and you'll probably get it again in a future session, but it's a really important topic. So we're going to dive a little bit deeper into that. And then transition into the carbon cycle in soils. And then the soil microbes' role in nutrient availability, and then how they're also influencing soil structure. So you'll see as we go through my talk, I've got lots of photos. I think photos, speak 1,000 words. So as I've seen things kind of out when I'm in farm fields that kind of are great examples of this good, healthy, functioning system. I've got photos of those. But then I'm also going to share some things that helped me with my understanding as I was kind of learning and on my journey as well through this. So organic matter, I often find as I'm having conversations with farmers, this is the topic that any farmer, no matter what crops they're growing at any scale, from a couple acres vegetables all the way up to 1,000 acres, everybody understands organic matter and the value of it, and they're all trying to kind of increase that organic matter levels, right? So sometimes this is a good kind of entry level point into a conversation with a farmer. And hopefully, as you guys are going through this course, and especially the soil biology course, you'll have kind of that knowledge a little more in depth probably than what a farm needs to know at this stage. But you getting this base knowledge and understanding will kind of help you explain things to the folks that you're working with. So on average, though, our nation's tillable soils have lost about 50% of their historic organic matter levels. So a lot of farmers that are coming into the system now, maybe taking over the farm, they're really farming a degraded system, but they don't recognize that, right? Because they don't know what those old historic levels have been in the past, right? So our long history of tillage has factored into this. Since World War II, the heavy use of kind of inorganic fertilizers has factored into that degradation as well. The other thing that it's pretty common knowledge is that organic matter acts as that sponge, right? So when we get those rains, when we're in those droughty periods and we get that rain, those soils are able to better hold on to that moisture, right? In that root zone where we need it. And I've seen this statistic kind of all over the board. But for every 1% increase in soil organic matter levels, soils can hold close to 18,000 gallons of water per acre. And like I said, I've heard this from the 17,900 that I've got here on the screen all the way up to 27,000 gallons per acre. So just take away that it's a large water holding capacity that we have with each increase in system. And then the other thing that, again, farmers really relate to is that drought resiliency, right? So as our weather patterns are changing, we're getting more fluctuations. When we've got higher organic matter level contents in our soils, we're seeing less fluctuation, and those crops can get through those dry periods better. Better soil structure, we're going to dive into that a little bit later. And then it's that critical source of organic nutrients, and it helps really buffer that soil pH in our systems. So traditionally, how did carbon get into the system, right? When we're encouraging management practices, historically, we often thought that leaving that above ground residue on the surface, those old crop residue, is how farms are going to build that organic matter levels in, right? So we got good heavy corn stalks here in this example. Wendy mentioned it's kind of that outer shell of the corn stalk that has the higher lignin content that can get incorporated and end up in that really long-term stable component of it. The other way that we're getting manure into, or carbon into the system, is manure. So this is one of my photos. This is a terrible spread pattern for manure. Not a great example of that, but it is a good illustration of that good, rich, carbon dense material and that carbon value that we are getting when we've got manure in the system, especially like a good bed pack manure from beef or bedded dairy cows. But what we're understanding now is this is not the only way we're getting carbon into the system. And actually, a lot of this above ground carbon that's left as residue doesn't actually make it into long-term stable organic matter. So what we're now hearing conversations about is this liquid carbon pathway. And this is when it takes a living plant in our soils going through that photosynthesis process, right? So back to your high school biology days, that photosynthesizing plant can take that carbon dioxide from the air, mix it with that sunlight energy and water, and create oxygen. And it's pumping a lot of those simple sugars that it's creating through that photosynthesis down into the root zone to build that root zone community, and then also leaking some of those simple sugars out as root exudates through the root systems into the soil profile itself to really help feed and drive this soil microbiome. So we used to really think that soils were a one-way pathway providing to the plant, right? Provided the water, provided the nutrients, gave that rooting structure. But we now know it's a two-way pathway, that living plant is feeding and doing just as much for that soil as the plant is extracting from the soil itself. So this carbon cycle, as you can see with this graphic and these arrows, it's a really complex, complicated system. And one of the challenges when we have these new soil carbon and paying farmers for carbon sequestration is it's really hard to really hone in on what part of that soil carbon sequestration ends up in this long-term stable humus component of the soil organic matter. Because as you can see, through all these different processes, a lot of that carbon that's captured or left on the surface as leaf litter, as it's broken down, gets respired and transformed as carbon dioxide, and actually will eventually leave that soil and get cycled back up into the atmosphere. So I have two stars on this graphic as well to kind of highlight there's really only those two input sources to get the carbon in. And first and foremost, it's going to be that living plant, that photosynthesizing plant to capture carbon. But then the other way is potentially those amendments, right? Adding manures to the system, some compost to the system, biochar. There's more conversations happening about biochar. But if you really go back to the root of all of those things I just listed, the compost, the biochar, and the manures, they're all plant-based materials as well. So ultimately, it's only that photosynthetic process that we're adding carbon into that system. So Fred Magdoff touched on this, right? I'm sure through this training and other education that you've had, you've seen this pie chart of the different components of a soil. And I'm not going to spend a lot of time on that. But this pie chart represents kind of a real, true, healthy soil structure where you do have almost half of that pore or that soil volume as pore space, either air or water pore space. But this tiny little slice of the pie is that organic matter levels. So one of the things that I found years ago that kind of helped me understand better organic matter, right? Because it's a really broad term, but as Wendy Sue explained, there's lots of different components to this general broad term under organic matter. So I came across this banking analogy that really helped me better understand. So I'm going to walk you through that a little bit here. So Fred last week said soil organic matter is like the living, the dead, and the really dead, right? So the living component of that would be like the bank tellers, or in this case, the soil microbes. So they are going to be the workers, right? So they're going to be taking the money in, the carbon in, they're consuming that carbon, they're processing it, and they're sharing out to other resources and other components of that soil profile and that soil community. So then we've got the checking account. So this accounts for that fresh residue that's in the surface. So that's going to be that fodder that's left on the soil surface. It's going to be dead roots that are slowly starting to decompose. It's going to be manures. So that's going to be that really fresh, active carbon. So that's like the checking account of the carbon. That's rapidly turning over, it's rapidly changing forms. We're getting deposits, but then there's carbon going right out of the system, right? So we all know how much activity we've got coming in and out of our checking accounts. And this checking account, part of that microbial food, is now able to be measured. A lot of the soil health tests do a water extractable carbon component as part of that soil health test. And that's part of this fresh residue microbial food carbon source that they're now starting to measure and report out on some of those soil health tests that the labs are doing. All right, so then we've got our savings account. So this is that decomposing organic matter, kind of the older than the fresh residue. So it's still pretty active, but it's where we start really putting some savings aside, right? So there's not as much activity in and out of this carbon pool. And this carbon pool is really what's starting to influence that physical soil structure. And then we transition to the retirement account, right? So this is that good, stable humus component of organic matter levels that have been chewed on and chewed on by those fungal components. And that's really where we get those nutrient holding components of soil organic matter, and that sponge water holding capacity is in that humus and that retirement component of that soil. So this takes a lot of time. So I like this image from North Dakota State University because we start with this really big pool of carbon as that kind of fresh residue litter layer. And as that residue starts breaking down over days or over months or over years, you can see at each phase of that, we've got these offshoot arrows of carbon dioxide that are leaving the system, right? And being respired back out into those soil pore spaces and eventually out into the atmosphere again. So a lot of that carbon that's left above the surface, almost as much as 85%, is going to get actually respired back off as carbon dioxide. So it's the bacteria and the fungus that are really efficient at keeping this carbon in the system. And the fungus especially, we need to have that in the system to digest those really long chain carbon compounds, those complex carbons, and also fats, right? So when we have a plant growing in a good, healthy system with some good excess nutrients, they can actually produce lipids and fats with that extra energy. And those lipophilic acids and those lipids actually contribute to the humus component as well. And then we've got this arrow coming from those soil organisms themselves, because as we're learning, also what contributes to that long-term stable humus are those dead cellular bodies of the bacteria, of the fungus, and those dead organisms are contributing to that humus labor as well. So if there's one thing that is worth your time and effort to kind of learn about is this carbon to nitrogen ratio, because you're going to be hearing it in a lot of different scenarios, one of which is residue breakdown, but another bit is going to be nutrient cycling that we'll touch on a little bit further. But I like this graphic because not all residue that's left on the surface is created equal, right? So depending on what the crop is, what growth stage that crop is, that carbon to nitrogen is going to be constantly shifting through that growing cycle of that plant. So typically, like you can see on the bottom of this chart, our legumes have a much lower carbon to nitrogen ratio. So they're going to decompose and be broken down much quicker. But as we're moving up in maturity, we get later rye that's getting reproductive stage, at like boot stage and heading out. Then we get a lot higher carbon to nitrogen ratio in those straws and that corn stover. So that is going to take a lot longer to break down. And that's when we need that fungus in the system to really break down that high carbon to nitrogen ratio. So just think about when you're walking a field that was in soybeans the year before that. Typically, if you're walking at harvest time, you're not going to see much of that soybean residue from the previous year's crop, right? Because it's that lower carbon to nitrogen ratio is broken down. But in contrast, if you're walking a field that had corn in it the prior season, in all likelihood, you're still going to be seeing that corn residue just because that carbon and nitrogen is so much higher. All right, so we've got fungus in the system that break down this carbon. Wendy Sue talked about that mycorrhizae fungal component that is really living beneath the surface, and it forms that association with the crop roots. What we're talking about here, typically at the soil surface, is a different family of fungus called the saprophytes. And the saprophytes are those real active decomposing fungus, and they reproduce with mushrooms. So anytime you're walking a farm field and you're seeing mushrooms in the system, you know that farm's got that good saprophytic fungus community happening. And you're probably, and the farm would probably say that, you know, they're getting that better residue breakdown in the surface. So that is definitely one thing that you guys can look for as you're walking the fields. Keep an eye out for those mushrooms. So worms also break down that residue. And again, Wendy Sue went into this. And worms are important into the system for a lot of different reasons. So in this graphic, we talk about 3 general families of worms. Those red wigglers, these up here, we don't often see them in an ag field setting. Most of what you're going to see when you're digging a hole out in the fields are these endogenic worms, right? These are the ones that live in that top 6 to 8 inches. They're going horizontally, they're going vertically, they're coming up to the surface and leaving these nice rich castings on the surface. Or we're going to have night crawlers in the system. And night crawlers can wreak some havoc if they're in a forested type system, but in general, in ag fields, we really like them. So night crawlers behave differently. They live their entire lives in the same vertical burrow. And these worms can live 5 to 7 years. So they're coming up at night, especially under moist conditions. They're coming up at night. They're gathering up that surface residue around their burrow, and then they're pulling that back down in. So one thing that you guys can keep an eye out for, and I always look for, this is one of the first things that I look for when I'm walking into a farm field, is how that residue is moving, and I'm looking for these night crawler holes. So this photo down here on the lower right of the screen are earthworm night crawler middens. So do you see these little clusters of this old, I think this was old soybean residue in this field. So if you were to actually kind of peel back that midden of crop residue, oftentimes you'll find like a pencil-sized hole, and that's that night crawler burrow. The other great thing that worms do in the system, right? They're creating that pore space, and especially in a no-till system, once this worm dies in a no-till system, that channel remains in place for up to 25 years. And those channels can go down till they get a restrictive layer, right? Either bedrock or maybe high water table. So I've done soil pits when I've been at field days, and sometimes you can find these old night crawler burrows with crop roots that year after year, those crop roots follow those old night crawler burrows down, right? Down to get those lower nutrient sources, those water sources. So especially in a no-till situation, these night crawler burrows are pretty critical, right? And also helping with that water infiltration. So again, how are plants going to sequester carbon as they're growing and dying? It takes that living plant system as many months out of the year. And this is really why we advocate for cover crops to be in the system, because it's not enough to feed these soils and these microbial communities just in that like 5-month summer growing season. We really need to get cover crops in there to extend that fall window. And then also to be there in those fields in the spring as the soils are warming up, right? So we want to be able to capture as much carbon throughout that growing cycle and throughout the year as much as possible. So those plants are putting a tremendous amount of energy and those simple sugars that they're capturing down into that root system. They're going to put a lot more of that carbon and root exudates out at the growth stage. So like in the corn framing stage, when it's really putting on a lot of top growth, it's pumping a lot of that carbon and simple sugars to feed that soil microbial system to get the nutrients in the water that it needs. And then typically as plants mature, they start shutting down and they're not putting as much of those root exudates down into the system at that point. So it fluctuates throughout the year, but there's some estimates that as much as 40% of the carbon and simple sugars that plant photosynthesizes ends up into the soil and into the root systems. So then after the crop is harvested, hopefully we got crop residue on top. Like I said, that does contribute to the carbon pool, but most of that is going to be cycled back off as carbon dioxide. But then we've got the roots in place, and they're slowly going to start decomposing, being that active carbon layer and adding to that pool as well. So I got this slide. Will Britton up at Woods End Lab in Maine shared this many years ago at one of his webinars, but it really kind of illustrates those carbon dioxide concentrations in the soil. Sorry, let me go back with that. So we've got a really high concentration of carbon dioxide in the soil itself. Right above that soil layer, right? Our concentrations then kind of diminish as we go up through that crop canopy. And we often don't talk about carbon being that big crop nutrient need, but it really is, right? And plants have evolved to get this soil carbon in the system. The stomata where the carbon comes in is on the underside of that leaf. So these plants are designed to be capturing that soil carbon in the system. And corn needs a tremendous amount of carbon in its growth. So I like this chart because oftentimes we ignore carbon as a major nutrient need for a good, healthy, functioning crop system. Oftentimes we focus on this NPK. I apologize for this, guys. My slides are automatically timed to start for 15 seconds. This NPK in the system, farmers do a really good job at replacing these big three, but there's this whole suite of micronutrients that crops need, right, at different growth stages, and they need to get that from the soil. So as we're building organic matter levels, we're able to capture and utilize more of those organic micronutrients that are in this system, and we're able to drive that soil biology that's going to go out and acquire some of those micronutrients. So a typical soil health or a soil test, you're going to come back with a cation exchange capacity, a CEC number on that soil test. And that's an ability of the soil to hold those positive cations, right? So I've got them listed there. I'm not going to read them, but a lot of those micronutrients have that positive charge. And that CEC is going to be determined by primarily the clay content of that soil. So clay has a lot higher ability to hold on to those cations, say, than a sandy soil, right? But that amount of organic matter in the system is also going to factor into that CEC value and that soil pH. But what's cool about that humus component of soil organic matter levels, it can hold both negative and positive charges. So especially when we're talking about nitrogen in the system, when that converts to the nitrate form, that's now negative. And typically that leaches out of our soil profile beneath our root zone if we've got some heavy rain periods. But when we build this organic matter level, that human has the ability to hold 5 times more nutrients than the clay components. And it can hold on to those negative charged nutrients as well. So plant roots, as a farmer's putting out fertilizer, right? He thinks he's fertilizing his crop, but plants are not very efficient at all at absorbing that nitrogen. They really have to kind of just passively come in contact with that nitrogen as they're growing and those root hairs are growing, right? They don't produce those enzymes. So Rick Mulvaney out of University of Illinois, his group is doing some really good research on this nitrogen use efficiency conversation, right? And they're finding that really a small percentage of that applied fertilizer nitrogen every season actually makes it into that growing corn, especially. They spend a lot of their efforts on corn nutrient efficiency. So they can do that N15 isotope markings, right, in the fertilizer, and then measure how much of that isotope actually makes it into the plant tissue itself. So they're finding that only about 30 to 45% at most of that applied nitrogen is actually making it into it. So bacteria is actually much better at absorbing that nitrogen than the crop roots themselves, right? So bacteria have enzymes that they can go out and convert those various forms of nitrogen. Urease is one of those ones, and they're just much more efficient. So if not much of our fertilizer is getting into our crop roots, then where's that coming from? Right. And that's coming from this organic matter pool. There's a tremendous amount of nutrients out there, but we need that mineralization. We need that microbial community to mineralize that. So if we really want our soils to be microbially active, we need to really keep those soil temps in a sweet spot for both temperature and moisture levels. So a lot of these microbes are going to go dormant over the winter. So as this time of the year, for sure, as soil temperatures are low, they go into that dormancy stage. And they're going to start waking up in the springtime and especially get really active starting around 60 degrees soil temperatures. So again, I mentioned earlier how important it is to have cover crops out there. So as those soils are warming up, and these microbes are getting active again, we want that living cover crop out there photosynthesizing and pumping these fresh sugars and root exudates down into the system to really start priming the pump before planting season comes along. So that activity is going to double for about every 10 degrees of soil warming that we have. And really that maximum mineralization is going to be when soils are between about 86 and 95 degrees. So it's really important to keep it below 95 degrees. When we get above 95 degrees, we actually start losing soil moisture through direct evaporation off the soil as well. So one of those practices, those soil health principles, right, is keeping that soil covered, keeping that good mulch layer. So that's going to provide that shading effect for the cooling, and it's also going to help with that moisture retention as well. So tillage is also really going to change the soil community as we go through. So, right, so we know that tillage is a big physical disturbance to the system. It's going to break down those soil aggregates. The other thing that tillage does is it's introducing oxygen deeper down into that soil profile than under that untilled situation. So as that oxygen is getting introduced deeper into the soil profile, it's stimulating this family of bacteria, the copotrophic bacteria. They respond really quickly to those increased oxygen levels. And bacteria have this amazing ability to just reproduce really quickly. So remember that carbon to nitrogen ratio, right? So as they're getting more active, they're doubling their populations, they're using some of that available nitrogen in the system, they need a carbon source. So they start consuming that carbon, that especially that fresh organic matter levels. And then tillage is also going to destroy that kind of spider web fungal network that Wendy Sue had some great photos on. And a lot of this carbon dioxide lost in the system actually happens in the first 5 hours after tillage. So they've done studies. You can see the bar graph here, but a full moldboard plow tillage event is going to have a lot more of that carbon dioxide escaping from the soil than the low disturb no-till type situation. All right, so I've said this a couple times, but bacteria are really what's utilizing that nitrogen in the system first. They're really efficient at it. But once they absorb it into their bodies, right, then it's good, stable nitrogen fertilizer. It's no longer at risk for leaching or volatilization. So it's not always a bad thing, right? And under those optimal conditions, bacteria can double their population in 15 to 30 minutes. So when we have a dry period and we get a rain and we get that flush of moisture back in the soil, they can really respond quickly and expand their populations. And that typical carbon to nitrogen ratio for the bacteria is about 5 to 1. And then we've also got this mycorrhizae system in place. And again, that expands that active root zone around that crop when they have this relationship with mycorrhizae fungi. And this mycorrhizae fungi can't produce its own food. So it relies wholly on that crop root to provide food, to provide those simple sugars, to feed and build its own network. And then in return, it's going out and scavenging water, scavenging those nutrients, those different micronutrients at different growth stages of the plants to bring it back to the crop root. So what we're also understanding in soil health is that plants are not absorbing only that nitrate and ammonium form of nitrogen, right? When I went through my nutrient management trainings years ago, the only thing we focused on was inorganic nitrogen. But what we're learning is that crops and plants have the ability to do these amino nitrogen. So they can access and utilize nitrogen that's already in these kind of complex carbon chains as amino acids, as amino sugars, and even kind of some of those complex carbon chains that are part of our microbial community as well. So mycorrhizae fungi is going out as all this organic matter is breaking down, getting these amino nitrogen compounds and transporting that through its system into those crop roots. So as much as 2/3 of the nitrogen absorption from plants can actually come from these amino forms of nitrogen. So this is a video that Wendy Sue had a lot of videos. I also like using videos because it's really hard to visualize these microscopic systems. So this is fungal filaments, hyphae, right, with bacteria moving along. And they say that this is in real time. And I apologize for a little bit of the blurriness. It'll get better as we zoom out. But you can see that fluid movement within the hyphae of the system. You can see the active, these fungal hyphae act as highways to transport these bacteria through the system. And then as we're zooming out, we're seeing that there's also stationary fixed nitrogen that live on these fungus hyphae as well, right? So these fungi hyphae, they can host phosphorus solubilizing bacteria that then are transporting phosphorus back to the soil. And all of this huge host of bacterial and systems that we have. So if we've got all this nitrogen that's tied up in our fungal communities, in our bacteria, it's not going to be plant available until it's mineralized, right? And Wendy Sue talked about those protozoa in the systems. So here's some scanning electron microscope images that get into a little bit more detail of what those protozoa look like. And if we don't have this predator population built up in the soils as well, we're not going to get that good nutrient cycling that we do. So here's one of those ones that are in those fixed locations, and they have these little hairs that kind of filter feed and bring stuff in. But then we also have the swimmers in the system as well. And then we've got nematodes that are also feeding on these as well and cycling. So again, just another quick little video of what these protozoa look like. And you can see inside the bacterial cells that they've got inside their systems, as well as they're grazing and moving through the system. So this is a slide that I often use to kind of, as I'm teaching farmers about mineralization, right? And this importance of having predators in the system. It goes back again to these carbon to nitrogen ratios. So protozoa are about 30 carbon to 1 nitrogen. So if their main prey are these bacteria with a much lower carbon to nitrogen ratio of 5 to 1, these predators, in this example, he's going to need to eat 6 bacteria just to meet his carbon needs, right? But in the process, he's also consuming a lot of extra nitrogen that it doesn't need to maintain its carbon to nitrogen ratio. So as it's accumulating that extra nitrogen, it's secreting it, right? It's like protozoa poop, and it's excreted in the ammonium form. And plants actually prefer to uptake inorganic nitrogen in the ammonium form. It costs them less energy than converting nitrate into proteins. So that's kind of that importance of that carbon to nitrogen ratio and that balance. And when we're talking about mineralizing nitrogen, that's what's happening in this system. So here's a couple examples of what you guys can look for out in the fields to try to judge how well these systems are functioning, right? So the best tool that you can have out there on every farm visit that you're doing, I want you to have a shovel with you. I use one of those kind of narrow spade style shovels that you can kind of dig deep, but also dig a pretty narrow hole. But that's going to be the best tool that you've got with you. Just go out and start digging some holes and looking at these soils. A good healthy soil system is going to look like cottage cheese, guys. It's going to be crumbly. It's going to be loose. It's going to be rich in organic matter levels. If you're digging up a soil and you've got this really blocky, plated, compacted soil with no structure, right? You know there's some room for improvement in however that farmer is managing that system, because that's not a real high functioning type system. Wendy Sue talked about the rhizosphere. So the rhizosheath is what is the visual portion of that. So as you're digging up plant roots, so another tool that I like to have, if you guys have your cameras on, I love these soil knives. They've got a serrated blade. They're kind of concave. But this is also a great tool to have out in the field to dig up some crop roots. And you can dig quite a bit of soil with that soil knife. But dig up some crop roots and look to see if you've got these aggregates hanging off. You've got this rhizosheath along this root zone, because most of the activity that we're talking about in this soil is happening right along that rhizosheath. So only like thickness of a penny about beyond that root zone is really active. And we're going to have like 2,000 to 5,000 more microbes in that real thin layer right along the root zone than you are in the rest of that soil profile. So dig up some root zones. Here's a picture in the lower right. This was only like 8-day-old rye seedlings. And you can see at a very young age, almost at germination, these plants start forming these associations with this microbial community. So oats, especially oats, are really good to have in the system. They're highly mycorrhizal. They really kind of change that redox potential of the soil, so you get better nutrient absorption. So anytime you're working with a farmer, encourage oats in the system as much as you can, or we're talking about a cover crop mixture. Really try to encourage oats, because I just keep learning so many positive things about how oats interact with the soil. So as we've got this good, healthy soil structure, we're not going to get that good cottage cheese crumbly soil without that good soil aggregation. So our soil microbial community is going to contribute to this in several ways. This middle picture here that looks like these kind of spaghetti strands, those hyphae from the mycorrhizae fungi will actually physically wrap themselves around the soil particles to create those aggregates. But then the bacteria and the mycorrhizae fungi, they create these biological glues, glomalin, right? And you can see through these red examples where these arrows are pointing, these filament kind of glue-like structures that are secreted to really create that good soil structure and earthworms castings. So this soil structure is also kind of another way for you to kind of enter into that soil health, healthy soil conversation when you're working with farmers, because farmers understand soil structure. It's usually something that they can visually see as they're kind of maybe transitioning to no-till or starting to incorporate cover crops. They can dig a hole. They can actually start to see this structure changing in their soil. And they'd want to avoid compaction, right? And especially when we're working with dairy farms have a lot of equipment passes over those fields, right? Chop and silage, they're getting manure out maybe a couple times a year. They've got heavy equipment, heavy trucks running on those fields, a lot more than just a row crop grain farmer does. So again, something that's very relatable to the farmers. The other thing that happens, when we're working in a more high tillage system, we're not keeping those soil covers, those soils are baking, we do get this soil crusting on the surface. And remember back to that carbon dioxide slide of the different concentrations, these soils need to breathe, right? So when we get this crusting on the surface, oftentimes we're concerned about it from water quality and we're getting more runoff to the field, but it is also that physical barrier for allowing these soils to breathe, right? So we need oxygen to get down into the soil profile. And sometimes crop root development to go live deeper onto the soil may actually be limited because there's not enough oxygen down into those lower soil profile levels. But in exchange, we also need that carbon dioxide through this respiration decomposition process to be able to leave that soil. And as long as we've got that living crop and plant above the soil, it's going to just cycle that carbon right back in through photosynthesis. I don't know if you guys can also see, this was out in the field. The farmer had just side dressed urea. So do you see all these little white urea pellets on the surface? So picture if we get a heavy thunderstorm coming through after he just side dressed, a lot of that with the soil crusting, that water's gonna just wash out that urea, and it's not even gonna get to the crop root zone, right? It's gonna end up in the surface water somewhere. So the soil structure, right, plays into that conversation of how water is going to flow across these farm fields and the influence that they have. So there's this concept of effective rainfall or non-effective rainfall. So in Pennsylvania, we get about 45 inches of precipitation a year, but depending on how that soil is managed, not all of that equal amount of rain is going to absorb into the farm fields on this side of the fence row as it may on the neighbor's fence row if they're managed completely different. So when we get these good, healthy, functioning systems, we get a lot more water infiltration. We get a lot less water runoff, a lot fewer erosion issues. And then we're not having that phosphorus loss to the system, right? As we're eroding that soil, we're not losing that precious topsoil layer and that microbiome that we've been trying to build with all these practices. This is an example that one of my dairy farmer friends took, which illustrates greatly just the power of management decisions on changing the way that water flows. So he stood on this paved road right where this yellow star is, the field, the bigger field on the right side of the road. So this picture on the right are his fields. He's been continuous no-till for about 40 years, cover crops for 20 years, and compare that to the neighbor that is growing some tobacco. They're doing some tillage. They're not very consistent with cover crops. So after this same storm event, same point in the landscape, same soils, you can see the difference with water laying in that farm field, right? And that's where we get problems occurring. So under these kind of ponding areas, when we don't have good drainage in the soil, you start changing the community as well in that soil. So after about 48 hours under those flooded conditions, those aerobic microbes are going to start to die off. And oftentimes when we're talking about the bad guys, right, these pathogenic bacteria and fungus, they prefer those anaerobic conditions. So if we can get better drainage in the soil, better soil structure, we're hopefully going to see the incidence of those disease pressures in our crops lessen over time. And then we don't want to lose that precious nitrogen that's in the soil. So a soil doesn't even have to be completely saturated for that denitrification process to start. Only about 60% of that pore space filled with water will start that denitrifying process and start volatilizing and losing that nitrogen from the system. So you guys may not always have the opportunity to be out there right after a rainstorm to kind of evaluate that. But oftentimes when you're walking a field, you can see signs in the field where those ponding areas happen, right? So here's an example. We've got a spot in this cornfield, right? Even from the road, you can see the color difference in this corn. But then when you actually get in the row, right, you can see the crusting and you can actually see algae growing on the soil surface. So if any of you guys have used that NRCS kind of soil health scorecard that they've developed, one of the questions on that is evidence of water ponding. So again, if you're not out doing that evaluation right after a storm, look for these other signs that you're going to see in the field to see if water's ponding and how that drainage is. All right, so all these things that we've been talking about, and I'm starting to wrap up. I see we're getting close to 4 o'clock here. Really what it comes down to at the farm level, right, is maintaining crop production, having that kind of consistent crop production year after year, and that good nitrogen use efficiency. And oftentimes, as farmers are kind of on this journey and transitioning, they may not think they're making much progress until we get kind of a good dry spell. And then oftentimes they see that maybe their corn isn't curling as much. They're not getting that kind of droughty, dry weather stress and those signs on their corns that maybe they are starting to see at their neighbors down the road who are still doing, you know, maybe some tillage and they don't have these cover crops in the system. So this is an example. Dave Brandt just recently passed away, but kind of one of the soil health pioneers out in central Ohio. And they had a big drought back in 2012. And he shared these pictures of his corn harvest after that droughty period compared to what his neighbors were getting in that system as well, right? So it takes times. You may not see these quick results with the system until sometimes farms need this kind of aha moment of these bad crop years, these droughty crop years to really kind of motivate them to make these changes. The other thing that's really critical to building these systems is that fourth soil health principle, right? About adding as much diversity into the system as you can. So this is actually one of Wendy Sue's slides that she shared with me, but we want to create this functional biodiversity on the farms as well. Farms 100 years ago used to be way more diverse than what we have now. When they farmed with horses, there was a lot more pasture. They were growing oats that were great for the system. They had pigs, they had chickens. These small farms had a lot of cropping diversity and animal diversity in the system. But as things have condensed and we've become specialized, right, we've lost that biodiversity in the system. So getting that below ground diversity in terms of crop diversity, reducing the tillage to leave those roots intact and keep that food in the system, increasing plant diversity, right? Different plants put out different root exudates and they host different species of mycorrhizae fungi and different species of bacteria. So the more diversity that we've got in the plant community in the system, the more microbial diversity that we're going to have. And there's a whole suite of practices that do that. And then there's this concept of creating these biological or ecological kind of islands. And leaving these hedgerows in place is a great refuge system. Here's an example on a lettuce farm where they're purposely planting alyssum, I believe it is, to encourage some predators to come into the system as well. So anytime we can kind of maintain those hedgerows or have kind of this intercropping type system to build that diversity in the system. This is a much harder sell when you're working with farmers to try to encourage them to do this. But it's really important to have a system. All right, so wrapping up, it takes time to build these systems. There's so many new frontiers that we're learning about that are going to influence these crop healths. As we're building these systems, we're giving those farmers confidence to maybe start backing off on some of those fertilizers and so some of those other input costs. And we need, our job as conservation professionals is trying to figure out first off, how to educate, right? Because if farmers understand how these soils function, I think they'll have a little more reason, right, to maybe get cover crops into the system and do some of these other practices. But we also need to figure out how to incentivize more growers to adopt these practices. So farms have done a great job for millennium, right, about managing their above ground herd. They've worked this system out really well. There's a lot of information. But what we need to get farmers aware of is they've also got this underground herd of animals on their farm that need their care and attention and need to be fed just as much as their above ground animals, because it's these below ground herd of animals that are critical to these high functioning soils and these highly productive cropping systems. Okay, so with that, I need a drink desperately. I'm going to stop sharing my screen, and we're pushing a little time for questions, Lauren. Sorry about that, but
That's okay. We've just got one question from you in the chat from Tom. Would you please confirm what you said about crops absorbing about 2/3 of their N as amino sugars?
yeah, so and I encourage you, Rick Mulvaney, University of Illinois, is doing a lot of that work. And it's that big missing components that haven't been taught in agronomy classes and these nutrient management classes. But yes, as more and more research is being done, we are seeing that a large portion of the nitrogen that's making it into our crops are coming in these amino nitrogen forms. Illinois, their soil nitrogen test can actually test for that amino nitrogen components in the soils. And then some of the other labs that are doing the soil health test, it's SLAN, SLAN, and that's the amino nitrogen portion of that soil profile and feeding the crops.
Wonderful, thanks. Yeah, Willie, you got a quick question?
Also like a protein test, ACE, ACE.
Yep.
And that one also, is that going to give a similar reading?
That's another test. Yep, the ACE protein test would capture that amino nitrogen component.
Yeah.
Yeah.
Just a quick addition to that is Dr. James White at Rutgers has been doing the rhizophagy, you know, the actually a kind of consumption of the bacteria, getting rid of their cell wall, consuming some of the nutrients, spitting them back out, the hair, the root hairs again, letting them kind of reassimilate themselves. I think that's fascinating too. We're just finding out how many ways nutrients are available to plants when biology is functioning in the soil for them.
Yes. I'm so glad you brought that up, Willie, because yes, that rhizophagy process, I encourage you to do a little homework and learn a little bit more about that. There's just so much to cover. Wendy, Sue, and I couldn't squeeze everything into this 2 hours.
You guys did amazing. Thank you so much for both presentations. Final slide for the day. If you can look in your chat, you have a link to the evaluation form from today. So especially if you had any questions that didn't get answered, please do put it in there. I am sharing the questions that get asked with presenters on certain topics in advance. So please keep telling us your questions. Additionally, once you're on campus today, you can see several different additional recommended resources from Wendy, Sue, and Lisa for today's lecture. If you want to check out some more things, there's plenty to learn about the living soil. Thank you for trying to squeeze it all in today. Our next class will be on Tuesday, January 9th. So after the new year, and we'll have Lindsay Rule talking about the Soil Health Assessment Toolbox. So different soil tests and many interpretations and how those are all functioning. Please do explore campus if you haven't yet. And the homework for next time is reading chapter 23 of Building Soils for Better Crops. How good are your soils? With that, I just want to thank Wendy, Sue, and Lisa again, and everyone for being here on Tuesday afternoon. And we look forward to seeing you all in a couple of weeks. Thank you, everyone.
Yeah. Thanks, everyone.
Bye, everyone. Thanks, Lauren.